from Rizal. He has received a gold medal as a best performer award for year 2017 at Kuching. And I'm happy to share that he's a proud alumni of our institute. With this brief introduction, I welcome Sachin. Now I request Professor Sonali sir to illustrate this movie.
Thank you, sir.
Now I request
Yeah. ಹಾಕೊಡಲ್ರಿ ಹಂಗೆ ಮಾಡಲೆ ಸೊ ಅದು ಸ್ಲೈಡ್ ಚೇಂಜ್ ಓಕೆ ಯಾ ರಿಬ್ಬನ್ ಓಕೆ Okay, so very good afternoon to everyone. So at first, it's really a great opportunity and also honor to present to yourself where you have studied. So I thank the Kelly College of Pharmacy, Hubli, Department of Chemistry, and also Khar University for giving me this opportunity to come in front of all of you and share few things which are very hot in the market right now. And I'm sure you will all take away a lot of good things by the end of session today. So I understand it's afternoon and you all have fed your stomach and the blood is flowing in the stomach and not in the brain. So you're all drowsy right now, isn't it? Huh? Because there was no gap. So if there was a gap, then the concentration. So just a joke apart. I'm sure you're all not going to yawn or you're not going to be bored in the next few 30 to 40 minutes what I'm going to speak, because it's a really very important topic that is required for all the faculty members, because there is a highly dynamic change in research going on every day. I'm saying every day and not every month or every year. So it's important for the faculties or these research scholars to update with these advances that are happening in the market. So I'm going to talk to you about a tool. So it's related to the theme. So molecular docking, you all heard a professor from IIT talking you how much important it is. So before the molecular docking, there are few things in the drug discovery. So I'm going a little bit behind, that is the base. So if I'm talking about the base, that means no complex structures and anything, very simple topic. So everyone will understand what I'm talking. So before I get into the, okay. So I get into the main topic of discussion today. Let me share a few things that we all should be proud of. One is to be part of this profession, that is pharmacy, because we are not just doing things, we are just saving millions of people. And I think the teachers over here, the professors are doing an excellent job because that's very important. So just a round of applause for all of you. So understanding how important we are, let us know that the pharmaceutical industry, let me tell you how important we are in terms of business perspective. So what you're doing and how much you're contributing to the better tomorrow in terms of the betterment of the mankind, and also in terms of generating revenues that is nothing but the GDP of the country. So Indian pharmacy is, it's the third most, not just the Indian, but the whole global, it's the third most valuable economy. And we all know, we have witnessed during the COVID time. So the global pharma is, is valued at 1.48 trillion. So 1.48 trillion in 22, and we are going to reach 1.9 by 2027. That is all because of the research and the molecules. So the Indian market right now is 50 billion and expected to reach 65 billion by end of 2024 and 130 billion by 2030. So just give you a realization how much 1 billion is. Recently we all witnessed Ambani is, having a lavish wedding. So they spent 1 billion of USD, money for the marriage. That's the thing which no one even dares to do that. So 50 billion is a very big amount. That matters a lot for the country growth. Okay, I have to stay near the slider. Okay, so since you've understood how big is our pharmaceutical industry, see, let me give you a quick introduction. This is very important. You all have to know, and you have to be proud that where India stands right now in terms of pharmaceuticals. So you all know India is known as the pharmacy of the world. That means we are producing a lot of quality medicines, and we are supplying to US, Europe, Australia, Japan. You name a country, we are there. That's the power of Indian pharmaceutical industry, especially at the generic sector. 50 billion, we are third biggest in the volume. World ranking in 14th in terms of value. Growth rate every year, Indian industry is taking 10 to 12%. Export market 24.6 billion. We're exporting to more than 200 countries. Domestic market, like you see, you buy medicines in India, it's valued at 24 billion. And import values and the biopharma. So pharmaceutical is divided into two, pharma and biopharma. So the biopharma is again 49.8. The COVID economy, we made around 7.7 billion worth of revenue. The bio IT and research, this is something where I'm going to talk now, is related to AI and how the AI is going to play a very vital role in research and discovery, as well as the everything related to AI. So 5 years before, or maybe 10 years before, the government is putting a lot of efforts, energy to make sure the funds that they release reaches to the universities or the colleges. So I think the websites are very clear. You get into the funding, you come to know what and how to get the funding. I just want to highlight this, the recent budget is in 22-23, almost like 3,000 crores has been allocated. So national health, so pharma is one department under that. That means you see so much of funding, and this is all, I would say, no collateral, just a free money, but you need to be very capable to get that money to the institute. So that I'm going to tell you how to do that. So the Department of Pharmaceutics, last budget for the medtech sector, 5,000 additional money is released just for the research. So the colleges can tie up with IIT, they can collaborate with, we have our own NIPER institution, government ICMR is there. So you can take, each institute can take up to 100 crores of money. So that's a very big amount, sir. So with this, introduction, let us understand very, how to say, very important thing. What's happening, in 2024. Before I talk about 2024, you see in, 2008, 9, 10. 2008 was when I passed out, I completed my degree, and I was not even aware of all these tools. Maybe Orkut was there, but we never heard of, Facebook. Android, I think Samsung has just introduced, and fintech industry, like we don't have ATM cards that time. There was a syndicate bank next to our college. Go there, right? So now everything can be done online, and it's a phone pay and pay ATM. So Apple, Twitter, Netflix, these are all the things we can't live today without them. And these are the apps. So that much these companies have dominated in a routine lifestyle. So that happened in 2008, 9, and 10. So you see that how much things are important. If we had a realization that these things are going to happen, but that's fine. I think let's see what is going to happen now. Okay. Okay. So what is going to deep dive in 22, 23, and now in 24? You might have been hearing about AI everywhere. So AI in WhatsApp, AI in that, AI in this, AI in marketing tools. AI is dominating. It has still not yet penetrated properly in your working, this thing, because there are many tools that are getting developed. So understanding, we don't have to be a coder, we don't have to be a developer. Okay, how you just have to know which are the tools that will help for your routine research work or your routine, the practice of teaching. So big data and data dynamics. These are the words you have to keep in your mind, because in the next 10 years they are going to be a really very important terminologies. Real data, RWD evidence. So what the big companies are doing, Geo is doing, they're collecting the data and they're creating the evidence. Okay, if I make this plan, who is going to buy? That is the importance of data. So now let me talk about the research data. You have patents, you have clinical data, everything is spread all across Google. It's unstructured. So that data is taken and you can make evidence of it. That is nothing but writing a grants and a proposal or research publication. So data science and analytics, again, Power BI is a tool that is used, and you can present a lot of beautiful papers. AI in machine learning, we heard Professor talking about. AI is the, how to say, it's the mother. Under that, machine learning and deep learning comes below that. So data-driven initiatives for drug development, manufacturing, digital transformation. Machine learning is introduced in manufacturing. So we will do a machine learning, they will code and they'll say, okay, this batch we have got an output of 92%, but like they have done like 10 to 15 batch. The machine learning will predict, okay, if I change my raw material percentage, what will be my output for the next batch? Might be 98%. That's what machine learning does. So adoption of data, IoT, cloud computing, AI, and this is, I think maybe after 2025, you'll see a lot of quantum AI. People are talking now. Biocon is started to do research on quantum AI. That is the next to AI. Quantum computing, is very difficult. So it is 100 or 200 times more faster than AI. So with this basic uh, understanding, let us quickly understand the definition of big data is defined as, I'm going to talk to you about the difference between big data, AI, and machine learning. So big data is nothing but you have a pile of pieces, that is the big data. What are you going to do with that big data? Or somebody is talking about a method development of so and so molecule, then you have to go dig and find, or there is, if your library is well organized, you can pick, and again you have to see through hundreds of pages where exactly the information is one. So that is big data. So then it's a difficult, that is a difficult to store and also cannot be handled manually with traditional database system. So artificial intelligence is often referred to as system itself, and it is a set of technologies implemented in a system to enable to learn, to reason, learn, and to act complex problem. So I told about machine learning. It will help you to improve your output. That's not AI. It's just an ML tool. But AI can tell you that, okay, if you use this kind of raw material, replace this, then your output may be 99%. That is the difference between AI and ML. So the AI is the mother. So then talking about the machine learning, it's a subset of AI and used to predict data for the future based on the applied input and past experience. So whenever I recently had been talking to some colleges and they're doing some good research using AI and ML. So they are developing a modules with the help of faculty. So there you need data. So the more the data, the better you can develop a model and you can get. So this is just till now what I've given is an introduction. So let us, here are some benefits of big data and artificial intelligence. So we saw understanding the definition. Let us know wider data ranges, analyzing, activating a wider range of unstructured and structured data source, faster decision making. As I said, the software can help you to extract any information, that too validated. It will tell you, okay, what you're searching or what you're thinking is right. Some validation. So you can make a faster decisions because it is, we cannot remember more than 1,000 items. So the data can do that. The big data, the AI can do that. So for efficiency, because the work that you're doing that takes 1 month of time, you can do it in 5 to 10 minutes. So your efficiency is improving. Especially I just mentioned increasing synthesis capability, patents, clinical operation efficiency, and also help to reduce cost. So time is money in the industry. Analytical integration. So these are some of the basic definitions or the benefits of big data, AI, and ML. So now next, this slide again, just for your awareness. As I said, most of my talk today is very basic and simple. It's just an awareness talk for all of you, what the industry, what is the whole world is talking right now. Drug discovery, as we heard, Professor talking about molecular modeling, and he also explained about the drug discovery. It takes 15 to 20 years. Using AI tools, you can reduce that time from 10 to 15 years. I guess if you are well trained, maybe in 5 years. You see, almost 7 to 8 years timeline you're cutting down with the application of AI in drug discovery. So that is the reason, there are so many companies. These are different softwares, AI software that are being used. So the use is different. Development, precise protein structure. So there are different stages in the drug development where the software is precisely designed. So AI startups, there are so much of companies. Boltzmann is one of our friend, and they're based from Bangalore. So they are doing a novel small molecule drug discovery startup. Molabs, this one. Cyber AIDD. Cyber AIDD is the tool that we are going to introduce, and my colleague Anil, he'll be talking about how the artificial intelligence tool can help for drug target identification, drug repurposing. So we'll just give a simple demo to understand. Okay. Okay. So these are again example. All the top MNCs in the world, Eli Lilly, Amgen, GSK. I just put the top companies. The list is very big. They all are. These are all different AI companies who are putting the tools at different stage of development. So the reason I'm trying to show you all this is, I said 2024 what's happening, but already many things are advanced. Industry is applying. So the faculty also need to know. I know I'm not saying you need to understand everything, but you have to have an idea what is happening in the AI and the drug discovery, especially the synthetic department. So how the big data works? It's simple. The tool now I'm going to introduce you is one of the big data tool is Pharmacodia Global. So aggregation of the system, data mining, and AI design. So what is done is we aggregate the system, what is required. We do a data mining and apply the AI modules, algorithms. So that will help to get the knowledge, information, intelligence, and these will help to improve the core factors of the production. Production in the sense here you can also consider the research. So if you are in your students or if yourself are interested in an research academia based projects, so you can involve in some of the first in class drugs using the big data. So from big data to usable data, that's the core technology. So I'll just get into a little more basics because my talk is all about basics. So what is drug R&D, like research and development? We all know that the basics of chemistry, biology, pharmacology, clinical, toxicology, pharma science, and patent. These are all things required for any product to hit into the market. So next, how the big data is going to help to make it more faster, because biology, chemistry, pharmacology. So you have to get into each phase and you have to study. But how the big data helps? You see, I'm just putting drug data. It is related to. So in the big data, all of this information is in one place. So you just click search and press enter. It will give you all the details related to drug, synthetic routes, what is the patents, clinical, pharmacology, like A to Z. You can identify targets. Maybe you're doing some for your students MPharm project. So it will help you to get idea what project I can select. The global approvals for the regulatory department, clinical trials, synthetic routes, and patents. So this is all about the literature, and the last point is very important. Okay. So I'm going to show you maybe 5 to 10 minutes live demo, maybe in another 15 minutes. Before that, I'll be introducing the platform. As I said, Pharmacodia Global. It's a big data. So how the big data is going to help you in your routine activities, in your routine research. I'll just give a basic lights for you all to understand. The source of literature, we all know that we use article, books, and journals. So the cons is collection of data, multiple sources, time consuming. But in present scenario, the data analysis platform with AI makes it easy to access this information. So that's the message from this slide. Then I've just tried to put searching on the internet, relying on source untrusted data, struggling to copy and download due to limited access, outdated. So what the big data can help you to do is it can get you all the information that you're searching in US, Europe, Google, MSN, Bing, or name whatever the information, open source data you have that can be available. So vetted global data, easily downloadable, and real-time information at a click of a button. So the intelligence data platform offers a comprehensive platform dedicated to pharmaceutical information and making it in the go to the research scientist and professional in the industry. So again, with this big data, as I said, the source of information we also collect from clinical data, synthetic routes. So this is an again. So now how the big data is going to help you is not just chemistry, not just pharmaceutics, but all the departments, because the information, if you are pharmaceutics people, if you're trying to develop some combination drug or want to see what is happening in US, find out to NLBA, so they can find all the information here. Intellectual property rights, IPR is very important in the research. So having an landscape of the patents is very important. No doubt Indian patents, WHO, WIPO, and EPO, there are so many sources, but all the patents finding related to particular molecule or particular target is very difficult. So that will be easy using the database, regulatory information. So landscape, as I told you, he was mentioning about one drug. So that itself is one target. So there can be multiple targets or multiple indications from single molecule. So with Pharmacodia, you can also access monograph and compare. Okay, so this slide again is, again I'm going to show competitive intelligence platform for research, CMC, learning, drug development, and medical device or information is also accessible in the big data. So again, as I said, the Pharmacodia Global helps insights for R&D, develop status, and promote the study, MPharm and PhD. For example, I'm going to take an example of JAK inhibitors. JAK inhibitors are the one like for immunodisorders, that is for the arthritis. So JAK inhibitors again have different indications. So maybe you're working on a project and you are only focusing on the arthritis, but it has other indications. So what I mean to say is having the knowledge about one particular target can help you to prepare alternate MPharm project for your students. So it can also have this indication, okay, let's use the same drug for different indications. So that will be one project. And the same chemistry team can do the development, validation, method development of that particular drug for that purpose. So these are the different ideas, because we have come with a very traditional mindset till date and trying to go through the literature and based on the information what we are reading in the articles. But these database, because it has AI application, it will help you to get lots of ideas what you can do, the research or the project that will be commercially viable in the market. So then, and also the like incubation centers, there are some incubation centers that Yale University is having. So they also need a lot of information and data that can be used. So in a nutshell, I want to get into the live demo because that's the most interesting part. So what all information you can find in the database, that is Pharmacodia Global. So here you can see you can get the information of the worldwide drugs, that is small molecules, biologicals, therapies, and vaccines. Patents, world, US, Europe, Japan, China recently, Korean patents also are listed. Then approvals. See since I worked in the industry for 12 years, and I also did my research, MPharm research in like industry joint project. but still there is a lot of gap between the academia and industry. And I have seen in the past 1 year, a lot of initiatives have been taken up by the institutions or the universities to bridge this gap, like doing research, doing joint development research. Recently, I was at IIT Bombay, so we were presenting the same information. Every professor there has a company, and they are making at least 10 to 12 crores of revenue every year. And we were, it was surprise, not too much to shock, but what they are doing, the difference I'm trying to see, they are trying to meet the industrial expectation. That's the basic difference. See, I understand, dear faculties, that we have a lot of other things to do, like preparation of papers and so much of information you spend time, and we put it as an unnecessary work. But this is a requirement to get that ranking. Otherwise, your salary is going to be the same. So it's all interconnected. So what I'm trying to say is, using of such data can, you don't have to sit for hours and check for the information and think what to do. It will help you to overcome that stress, because you're getting all the data in hand, and that too you can print and do whatever. It's all depending on. So the IIT professors, what they are doing is, they are working majorly on the biopharma, because the bioscience department. So they have developed a simple upstream and downstream process. They have modified the production capacity rates. As Professor Wangikar, I was talking to him. So he has made a, just simply identified, he has given that project to all his PhD students, MPharm. He has validated all the system. Then he registered a startup in IIT only, and commercial aspects have started. It will take time. So that is very important. I think this is a simple thing. MIT, US, you go to Europe, this is what they do. I mean, that's the basic difference I'm seeing in the Indian. So there is a little bit of efforts is required from you all, because I know, especially the men, okay, we understand women are occupied with hundreds of things, and it's managing the house, office, and lots of things. So if you focus yourself what you want to do, I think that will give you a direction. Okay, I want to develop this project, or first year MPharm, you give this project a continuation to the second student. So that will help you to continue, tag you in the research pipeline. And one day, like we are one company, what we are doing is, if you develop something, we make your project commercial in the market, because we have been traveling all over India and China for a couple of years. So that we can help to do. So this is what I'm trying to say is, try to think, because the research initiation or the research interest is very low, and the paper also has been published recently, that even students are losing interest. If teachers lose interest, students definitely will lose the interest. So there have to be an ecosystem for you to get active. So get into these kind of tools. And also, like, the students having exposed and doing this thing, and they will get a very good pay jobs and salaries in the industry. So that will then, there are a lot of advantages. So let me come back to the database. So clinical trial data. So even the clinical department, PharmD, they can check all the information, because there are a lot of data is coming out every day, the endpoint results. So this will help them for routine practice, CMC, synthetic routes. We have a lot of synthesis routes present in the database that can help you to redo in the lab. And regulatory information, data service, consulting, and sales. So we also put some sales data. So what I'm saying is, having such tools, you can also generate revenue for your institution. And the moment you generate revenue, you are identified. That might be an advantage for you for any of the reasons. So this is about the Pharmacodia database. So try to know each project, try to make it more commercially viable. I'm not saying 100%, because research always has a success rate. So getting to higher success rates, know what is happening in the market, that through the database you can come to know each track, know the whole industry. So this is what the database can play a major role, way ahead down the line. So the Pharmacodia has been cited by many international journals. As I said, many of them have written overview, review articles, short communications, writing research grants, and a lot of things have been done using the Pharmacopoeia Global Database. So there are some books that have taken references, especially for the CAR-T therapy, because we cover what to say, the biological information. I'll get into the database demo. I know we are running late, so I've tried my best to cut short some of the information, but the demo part is important. Um, Now it's a simple web-based. You just have to put username and password, and then you're in. That's it. And this is how it looks. So I will show you the demo later. Let me jump to some more important things. So synthetic routes you can find, global information, sales data. So I have a lot of examples, but for today, I want to give you a simple case example. There are some colleges in Mumbai and also in Hyderabad where they are writing some consulting reports for the industry, and they're getting well paid for each report, 25 to 50,000 range. That's the revenue. So it can be done as a standalone, or it can be done through the institute. So this is one case example what you can do using the databases. One here is azilsartan. I know it is used for hypertension. It is a new molecule. So I have listed first generation, second generation. These all data I have extracted from the database. So I have put losartan, is the latest, which is giving good efficacy. So just I have done a comparison using our data. This data I have extracted from the data. So the uh, efficacy is 9.1, and the response and the side effects are comparatively lesser compared to losartan. So alisartan, sorry, my pronunciation, alisartan isoproxil. So this is much better drug in terms of efficacy. So you already know that, okay, it is good. So how about the structure? You can do the comparison, what is changed and what. So you'll come to know that. So I made one more. This is the simple case study I prepared using the database. So here, just a double phase evaluation. Right from 1995 to 2019, I have put all the molecules under the same category, which are being introduced in the market. So as a chemist, you can better understand what are the structural or the SAR modification that is being done that is giving a better result. So I'm just looking at the commercial aspect, value, high value and low value, because from here to here, a lot of molecules are introduced. So the value of unit dose has gone down. Even though the value has gone down, the volume is high because it's hypertension. Everyone is getting, this is a chronic disorders. So here I made a simple sales. I have taken China as a case because it's a very big market. So I have seen here telmisartan, candisartan, and azilsartan is somewhere like 0.03% sales. I have seen when it was launched in 2017, 0.26%. So you can see there from 2016 to 17, there is a increase in the sales. So this is how industry do is, okay, I am taking telmisartan is good, everyone is taking, but for the industry, because they are looking for the market for the next 5 years, and they are looking for the revenue. So this gives a clear indication from 2012 to 17 is increased. So this molecule is very good to put in my pipeline. So the pipeline due diligence we call, that is how they select the molecule in the industry, so that they start developing. So once they start developing, I think you know the whole process, starting from synthetic there are ready-made raw material suppliers, do it in the formulation, you have to go to the patents, clinical data, then you come up with the prototype product, then you go for tech transfer, scale up and commercial validation, then into the market. So this is the simple process, but there are a lot of things involved. How you get to know is through the data. And this one I just prepared a simple chart. So IIT Bombay company called as ImmunoAct. ImmunoAct is the first Indian company to develop a CAR-T therapy. So I'm sure most of you know what is CAR-T therapy, chimeric T cell response. So these are the cells which come out when they found any immuno disorder or any foreign bodies. So they take the blood CAR T therapies are like giving 90 to 95% assurance that you will be cured from the cancer. We are taking chemotherapy medicine. So these drugs are available in different world. But India, last 8 months back, we got the first approval. That's why, anyone, big people, big shots, they go to America, Singapore to get treatment because these therapies are available there. Now it is available in India. See the cost. 373, I mean, almost like 3 crores, more than that, for one-time treatment. Kimra, Escata, Ecras. So these are just, I made a comparison. Now I think there are 3 more companies approved in the last 3 months. So what is the CAR T therapy? This can be a simple short communication or review article. You can publish in any of the journal or anything. So that will give also a recognition for your research. So India, what they did is they developed CAR T for sickle cell anemia, and now it can be treated. Now the treatment is available in more than I think 20 hospitals all over India. Do you know what is the cost? Just 30 lakhs. Just 30 lakhs. Where is 3 crores? Where is 30 lakhs? That is what India has achieved in reducing the price. India is known for reducing the cost of the price. So if you have to get into that, so these are the basic. It all starts from you. So I'm just trying to put this. So that is about the the database and this thing. Just as I told, it's a basic thing. I just wanted to highlight. This is a tool that can help you for your routine work. So if you have any questions, I'll take it. Then I will just go with the demo, please. Okay. Sure, let's get into the demo. Actually, I wanted to give you all a free trial, open up and try to make a workshop kind of thing. But due to short of time, I'll just get directly show the demo. Later, I will share you on the free trial codes will be there, so they can get shared. So, how many of you are using laptops? So, can you get into this website or? Wi-Fi connection is there? Okay, so never mind. It's a good luck or bad luck, I don't know. But I'll give you the good thing is I'll still share you the free codes you can activate. You can use it later. So let me show you. See, this is how it looks. It's a simple website. So now what I have done here is, wait a minute. Let me try to sign out and show you how to log in for a free trial. So here you can see there is a register. When you get in, click here, and here it will welcome to Pharmacodia. So here you put in email ID. That's it, and then get code. So once you to the email ID, you'll get a code. It will be a 6-digit code. So once you get that code, no need to fill these details. If you have time, you fill it. Otherwise, just the email ID is enough. Okay. So then you put create an account. So this page will move out. So then you have to put sign in. So once you go sign in, you have to put that email ID and no need to put the code that you have, okay. Just you put your email ID and just type 123456. Remember, 123456. Like for example, okay. So here you have to put 123456 and just put sign in. So it will sign in, and then you see these are not highlighted. So if you click here, it is showing X. So this will come. So now here you have to use the invitation code. The invitation code which is sent to all of you. So I have put more than 25. You can use any one of them. Just put that invitation code, put the active. So remember, the code which is sent has to be put when you log in, put 1, 2, 3, 4, 5, 6, then put it here. So then it will activate for 3 days from the day you are doing activation. Let me get into my full paid account. So I have successfully logged in. So here you see all the active. So I'll just show you. W drugs refers for worldwide drugs, and R&D gate refers to the information which is relevant for your routine research. So as I said, all the departments can use this, starting from chemistry, SUTEX, QA, QC, analytical, pharmaceutical analytical chemistry, and other this clinical pharmacology or the PharmD students. So here you see in W drugs, small molecules, biological, cell therapy, and vaccines. Landscapes, that is majorly it is used during the drug discovery. So you need to identify targets, and license in the license or the sorry. So this is used especially for the pharma MBA to see the commercial aspects. So in R&D, you can see here, you can get all the information from US, Europe, Japan, China. So one more important thing I need to highlight here is we say we are a global data plus one. So global means US, Europe, Japan, Australia, plus one is China. So no database in the whole world is giving the deep information about China where we are. It relates to the approval, it relates to the patent, it relates to the clinical trial. Why China? If you Google out, the number of research publications are highest in China. Second, the number of patents are highest in China. Third, in the past 20 years, if you see, there were hardly few countable molecules for drug discovery in China. Now they are leading, they are dominating over the US. In 20 years, they have emerged. So there is so much of information and data of the molecules is there. So that data is here. You can use it for your routine. So here, this clinical NCT refers to as CTR for China. Then patents. Patent, as I said, world patents, US, EP, JP, and China. Even we have a South Korean patents. Once you search it, you will get it. So you can find any patents. You don't need to get into Google patents, anything. It is all available in one shot, all the different countries. So sales, again, it is, we did it for the industry purpose, but as I told you, Ali certain, if you want to do some commercial reports, you can do it, the sales. CMC, active ingredients, information, key intermediates, impurities, synthetic routes. Right now, we have not, when we are trying our best every day to make it at least 90%, but we have reached to 75 to 80% of all the molecules, synthetic routes in this. Formulation prescription, you can find like any molecule, you put it and it can show you what are the excipients they have used. So it will be useful for SOTEX people. So excipients, NMPA refers to China FDA. So Japan registration, that is the DMF. CEP refers to Europe. US DMF, you can check the DMF data. Korean DMF. Pharmacopoeias, yes, we also have pharmacopoeias. Like I'll just show it to you. This is also a very important tool, sub tool that we have put it here. So pharmacopoeias, it will give you EP, BP, USP, CP. IP we don't have. Very soon we are going to put IP because IP is accessible for all the colleges. So you can see here EP, BP. So you just have to put here the name of the molecule, or if you're only looking for USP, you put it. It will give you the monograph details. So dissolution data. Mostly US FDA recommends the dissolution data. So you can also find easily here reference. BCS classification is very important for any research because if it is class 2, class 4, you need to go for solubility enhancement. So you can find what is your BCS classification. So let me, 5 minutes I'm going to take and I'm going to end this demo. So I'm just going to W drugs. So how you're going to search, I'm going to tell you. In the drugs name, if you know, you can put target. If you know, you can put indication you're looking for diabetes, you can put, or you want to search by the company, how many molecules they have approved in the market, that also you can find. So, and if you don't know anything, you can do a structure search, exact or a similar one. So you search engine is, so I'll take an example of tofacitinib. Okay, see, once I type it will show you different short form. So just click and you see here 64,000 molecules data. When you search, the AI, what it will do is, it will try to get all the information related to 6 molecules. So if you see here, tofacitinib, it's plain citrate. Here is fingolimide with tofacitinib. I'm more focused on the tofacitinib plain. So it is approved in US 2012, in China 2017. It's a tyrosine kinase. So it has been, it's classified as first in class. That means in that category, it's the first drug in the market. So you can pick up this molecule for the MPharm or PhD work, because very little work has been done on this molecule. That is the first in class priority review. So I'll just put here AI recommendation. I'll just click here. This is one of the beautiful in the pharmacodia. See, you see targets JAK1, 2, 3. So under JAK1, there are 70 similar related drugs. Tofacitinib is one drug. The same indication, you can find 70 molecules information, and that too just in a click. If you want to find, search it through, online, it will, you can find it, but it may take a couple of hours. So here in just a second, and you can take, so indications, so it is having different indications. So that also you can find, and which are companies which are developing this product. So let me go back. One second. Okay. So I'm going to go with click here. Before I go there, let me. Okay. Okay. So search, then you go here and then you click here. If you click here, here it comes. Why the name Pharmacodia Global? Because it's a pharmacopoeia. Because we have end-to-end information of a particular molecule. General information, drug name, classification, physicochemical properties, all you can find here, A to Z. Then R&D review, like what is research is going on this particular molecule, what is the development status, means who have developed it and who have approved. You can see it is approved in US, it is approved in Japan in 2013, and China and Europe. So these things, and also what indication it is approved. You can see here, spondylitis, arthritis, rheumatoid. So phase 1, phase 2, phase 3. So it is all approved till psoriatic arthritis, till clinical. The red dot indicates these are the indication approved in the China market. So this will give you under, so you can go to the labels, then global approvals, as I said, it is approved in US. You get into FDA, you will find again the FDA whole details. There are thousands of documents you can check. Assessment, global sales, synthetic routes. Let me get into the synthetic routes. You see here, there are different tables here. What you want, you can check. So this is a synthetic route, route 1, route 2, route 3. So it will give you all the intermediates also used. You can also click and check their details. And we have given reference for each and every synthetic route. So you can also cross verify that. Then preclinical data, clinical data, you see, like there are 199 clinical data for particular to tofacitinib. So that information you can write a review article on the clinical overview of tofacitinib using 199 clinical data. So that can be, it can be published in elsewhere. Most of the people who are using have published in elsewhere. Then patents, can you see here? If I go here, see here, there are around 1,309 patents only for one molecule. So it's crazy to check all the patents. So I am a formulation scientist, so I am more concerned about the patents related to formulation. So here you see there are a lot of filters. So I'm going to put the technical type. preparation compound. So I'm more concerned about related to formulation. So 54, just apply. And these are all the global patents in one place. So it makes easy to find the patent. So here you can see there's valid, the original link you can download. So this is, I'll just stop it here because there are a lot of things that can be done using this database. So the free trial is there. Try to use it when you're ready to use it, because once you open it is valid for 3 days. And if you need more, you can contact us. And so this is about the big data. So just to conclusion, I just tried to try my best to put in your mind that the tools like this big data tools, there are other companies are also there. Try to use those tools to use for your basic research, because the patience level nowadays is very low for most of the researchers. That is why Instagram, even the YouTube has started the shorts, Facebook has started shorts, because everyone want to, in 10 minutes they want to see 10 information. So that is why these tools can also help you to get information. Unless you have more patience, then I think you can do some groundbreaking research at the institution we are in. With this, thank you so much for all of your time. And next, my colleague Anil, he's going to talk the next. The database is the base, so he's going to talk about artificial intelligence drug discovery, which is a very interesting topic. Thank you so much. Okay.
Thank you, Sutin, for giving the knowledge regarding how these big data tools can be used in the research, that is to accelerate the research rate, and also providing the detailed information regarding the pharmacopoeia. I believe that the participants will make use of the course what we are given, and we will explore the pharmacopoeia to the best. Now I call upon our HOD, Dr. P. M. Rohan sir, to illustrate the statement.
For the opportunity. Thank you.
It's a short term.
Yeah, no. Thank you. Presentation on the
So now we are having one more speaker, that is Mr. Anil Kumar Randel. He has completed his B.Com from our college. Please come. So Mr. Anil Kumar Randel is a student of our college. He has completed his B.Com from our college and a M.Pharm from HCL college. And he is a member of FIP as well as ITA. He has more than 13 years of experience in analytical research and development and analytical support for pharmaceutical development products. He has worked with Apotex India, then Biocon, then TV Pharma China, and CQ Pharma China. So, coming to his achievements, he has received Yanzhou Friendship Award 2020, the Forex Expert Award from Hebei Provincial Government of China for contribution in socio-economic and health and scientific development of Hebei province. He is now the in-charge and SME for Cyber Aid tool, AI-enabled early phase drug discovery tool. He has received a recognition award in 2019 from Hebei TV Pharma Company Limited for contribution to the organizational growth. So presently, he is working as Chief Scientific Officer for Suspinjara Life Sciences Private Limited, India. So we are happy to have Mr. Anil Kumar Randev for this session. So I invite our staff, that is Ronald sir. I request Ronald sir to welcome Anil Kumar. I request Anil Kumar Randev to take up the session.
Some pointer. ಒಂದು ನಿಮಿಷ ಇದು ಡಿಸ್ಪ್ಲೇ ಮಾಡ್ಕೊಡ್ತೀನಿ. Very good evening to all the participants, and my heartfelt gratitude to all my teachers. It's a, like, teachers are the sorry. So it is said that teachers are the path, and so I am very I feel very humbled and my heartfelt gratitude to all my teachers that they have showed us the path to walk, and later the path itself became our teacher. So again, my thanks to all my teachers, and thanks for giving the opportunity to introduce a new tool. Till now we have discussed about importance of structured data, what is AI, and how important is the structured data for the AI. So you can say the structured data is the backbone for the AI. So if we want to teach machine, so the data should be in the structured form. So that is what we have seen last 1 hour. So we have seen so many data which is in the structured form and machine learnable form. So once that deep learning, machine learning. So once that machine learns, so then comes the prediction. Machine starts predicting. So that a practical tool I want to introduce, a predictive tool, a generative tool, you can say, especially for the early phase drug discovery. So like this afternoon we have seen from professor from IIT. So he was saying like 10 to the power 64 of molecules. If you want to conclude like couple of like 200 or 100 molecules, that is by traditional way it is, I think, impossible. But that can be simplified using the AI technology. So that for that it is a practical tool called Cyber AIDD, the AIDD tool for drug target selection and molecular design for the early phase. So this tool helps to boost the efficiency. I think it's not that we are not doing the early phase drug discovery, but how efficiently we want to do it with a short time and very less resources. That is what this tool is all about. So increase the efficiency in screening the innovative drug targets and designing the molecular structures for the early phase drug discovery. Yeah. So what insights we are going to get by end of this session is global biomedical artificial intelligence market development trend. So how the market is developing, and AI technology applications. Some examples I will show, like which are already the work is ongoing using the AI technology, and case studies of molecular design, cyber CPT. I told creative models, that is creative. We already are, some people, I think we are using the chat GPT. Chat GPT is nothing but generative pre-trained transformer. Means we are teaching the machine with our language, like maybe the English or whatever the language, so that machine is generating content for us. Whatever the content, you maybe type, like write an email or leave, it will give you. So that is GPT. Ours is a creative pre-trained transfer model. So cyber SAR case studies also, we'll see a few 3 to 4 examples, and I will introduce the cyber AIDD platform. So cyber AIDD platform, don't think it's like a very complex science or something like that, because the IT part, the IT people will take care. So we are the users, end users. So it is divided into 2 parts. One is cyber SAR part, that is knowledge database tool, and second part is cybercom, that is discovery models. So it is very simple, like simple dashboard. There are 2 parts: cyber SAR, that is structural activity relationship part, and there is discovery part. So in the cyber SAR part, so like target, there are again some 4 sections in that, where we can explore the new targets, which facilitate the proactive exploration of potential drug targets. There are n number of targets, because as I told, we have so much data. Research has been done already, so there is so much data. So we have so many targets to explore, so that all exploration possibilities is possible using the CyberSAR tool. Then helps to identifying the hit compounds and provide the insight for designers first-in-class drugs. So for students and as well as the research scholars, so it gives the possibility or inspiration to explore the new targets and design the new first-in-class drugs. Third function of CyberSAR is to facilitate the rapid assay design, because a lot of assays has been reported already in the literature, so that all data has been is there in this database, tool, so that we can have a new assay design. Further function is efficiently acquire relevant information with the online survey and molecular design. So again in molecular cybercom part, there are 3 parts: like cyber hit, cyber lead, and cyber candidate. So in hit, like we can come up with the screening of around 200 molecules. Further, in the lead candidate, we may come up with the 5 molecules, and finally our main candidate. So I'll just start with the. Before that, I think this already a professor from IIT has told. So what is the pain point with the traditional drug discovery? So it takes again a lot of time, like and lot of money, but and also the success, like failure rate is very high, just 5% success rate. So not any small companies can take chance to develop a new drug molecules. All big companies, they have a lot of budget to do that. But what is the solution for that? So business models are shifting towards the cooperative models. So as you have seen in the earlier presentation, a lot of companies now they are collaborating with the AI companies to save that resources and money in the early phase drug discovery. So once they finalize the candidate using this all the tools, whatever it may be, like AIDD tool or computer CIDD tool or molecular dynamics tool. So using these tools, reduce those early phase drug discovery cost and use that like with a short time to come up with your lead compound. So this is one of the solution, and data-driven AIDD technology has become a new technology and methodology in the early phase discovery phase. So new treatment modalities are emerging. So that is drug repurposing. So a lot of companies in India, like we are visiting many companies for our business work. So a lot of companies, they are focusing their trend on drug repurposing, because repurposing means existing molecule, approved one, used for the new indication. So there is no need of doing the safety study again. Just efficacy is enough. So this is also one of the new very emerging field. So global biomedical artificial intelligence market development trend. So you can see like from 2021 to 2030, the from 1,400 to 2,100 billion dollar expected increment. This is the trend. So this will, I think, this will give the idea. So how these tools are contributing for the faster drug development process. So this is a trend, how the companies are using the AIDD tools for developing the new molecules. So this is, I think, from 2017 to 21 is there. So it is, I think, many folds it will going to increase in next few years. So this is the examples, like many leading multinational pharmaceutical companies actively investing in the AI. So you can see the examples, like any big companies, they are collaborating with the AI companies for the development, and you can see that even the details of deals what happened is mentioned here. So just this for giving you the picture that where will be our pharma research sector in next few years. So again, as I told, it is not started now. Already there are so many molecules which have been developed using AI, and they are in the clinical phase. So these are some examples in the oncology field. They're in the phase 1, and in neurology field, they're already in the phase 1, phase 2. So any like you can say, take any examples. So many examples you can just find in the literature that there are many molecules which are already have been developed using the AI, and they're in the clinical phase. And I think next maybe 1 or 2 years, the successful molecules will come out into the market using the AI. So this will become very important, especially when we are doing the post graduation or PhD, to know this information, and where should be our what should be our research area, and even for the you can, like, some companies, they are coming up with without having any lab. They just have the computer. They use this tool and they come up with some molecules and do the patent. And after 2 years, this patent is sellable to some big company. So that is the trend. So I think that is possible with even even at a student age. So this is the possibility what we should explore. So as I told, data depth calibration and the application of AI technology significantly improves the efficiency of innovative drug development. So how much significantly means? Around 70 to 80%. So it will reduce your time and cost. So these are some few examples. One of the example that one AI company, it has filed a patent. This is a patent number. So KAT6A, this is an enzyme inhibitory activity. So further improved selectivity and significantly enhanced permeability and kinetic solubility. So I think for KAT6A, for this enzyme inhibitor activity, there are so many molecules which have been already known. So analyzing these molecules and using the AI DD platform, we can predict like what side chain, or it may be the fragment replacement, can do improve what kind of it may be the improved efficiency, or like selectivity or druggability, or improved physicochemical property. It may be solubility, or it may be the other like permeability. So this already this I told, right? So many AI companies, they are working with this model. So that is possible, as I told, with like research universities and college level also. So this is another example, like compound with the clinically developed GLP-1R oral small molecule agonist, that is the danuglipron. The compound designed by AI company showed a significantly improved pharmacokinetic properties in the preclinical animal model. So it is when they came up with the improved version of the Danoclip Pro using AI model. So in the animal studies already it has shown improved pharmacokinetic activities. So I think this can be a project for our Pharma, that we can come up with our own molecules, and pharmacological screening can be done in our college level, and it can be patentable, and it can be like of commercial value. So few more examples, like from the patent analysis of SSTR4 agonist for the treatment of pain, like pain management molecule, a domestic AI company further optimized the stability of this liver macrosomal oral viability and solubility on the basis of original molecular structure. So this is like what possible. Just I am telling about the patent analysis. There is a lot of possibility, like using the SAR analysis and using the bioassay analysis. So we can come up with our own molecules which are patentable. So this is one more example that you can see. Just see these are all the AI companies which are filing the patents, not the pharmaceutical companies. So later they will once the preclinical or animal studies completed and the molecule shows the bioactivity, they will select for the big companies or they collaborate. So as I told, GPT, I explained, this is I think you are using that chat GPT and all. So here we are using the human language, and it will be like for the especially used for the content generation. But our model is CPT, creative pretrained model. So here we are using the chemical language, that is, and biological language, protein sequence, and so this when undergoes the deep annotation and transform annotation. This is the like IT section, how they are doing the deep learning, and so we get the innovation and new molecular structure, that is creative pretrained transformer. So now I think you have seen the background. So now I will introduce the how the cyber AIDD looks. It is a very simple dashboard, like even a first year graduation student can understand this. Not a very complex one, like which you have seen in the afternoon, that molecular dynamics and all. Very simple web-based login, and easy to understand by anybody. So as I told, this is AI empowered, because AI is analyzing the structured data and it is interactive. So chemist, his knowledge and the structured data, so it is interactive. So chemist can use his experience and knowledge also to input into this. And it's like first in class drug discovery and design platform for medicine chemistry. So totally three core functions are there for this. One is provides opportunity for drug discovering the potential first-in-class targets. As I mentioned earlier, first-in-class targets can be discovered, and efficiently conducting the SAR analysis. So this is a very, I think we already have so many softwares to do, like in the public domain to do the QSAR analysis. But the specialty or the uniqueness of our platform is like this SAR is based on the real-time data, means real-time structures, the structures which have been either in the clinical phase or in the approved phase. So that gives, I think, more confidence. Already your molecules are approved. So based on those approved SAR, you are doing your new molecular SAR. So there is a chance of failure is, I think, nil, I can say. So that is a uniqueness. The innovative structural core and lead molecular optimization. This is, I told, the three models you can run and find the new molecule. So advantages of the platform. So based on the data which we are talking, so this is based on 1 lakh plus targets and 2.43 million real active compound structures. So whatever data, what structure against which we are our AI is working is 2.4 plus billion molecules which are either in the clinical stage or in the approved stage. So that is the uniqueness of our product. Then 90 million activity data in the journals which has been published in the reputed journals is included in this. and 1 million documents and patents. So there is a lot of structural information even from the patents that also has been considered. So 10 years of data accumulation and validation of drug data covering. So as my colleague Mr. Sachin has introduced the pharmacopoeia database, that also has, I think it is a more than 10 years old database. So it has the structural information of 10 years that also has been considered for this AI tool. Then highly interactive. So as I told, chemist, it's not like you are working with not without interacting. So you can use your experience and your ideas to input into the tool to get the results along with the tools output. Highly interactive, simple and friendly operation interface. As I told, even the first year reformer candidate can understand the dashboard how to use. So again, I refresh. So CyberSAR, it has a tool can be of two major divisions. One is CyberSAR and CyberCom. CyberSAR is a knowledge database tool, and CyberCom it is a molecular generation and evaluation tool. And CyberCom again has CyberHit, CyberLead, and CyberCandidate. So I think till now whatever I explained, is there any doubts in this? Yeah, there is nothing like as I told. It's like structured data we have from last so many years, whatever the information. Based on that, the AI is predicting. Very simple. So we'll see the first one, that's CyberSAR. So CyberSAR is a target screening and latest reported structure activity relationship analysis. So as I told, it's quite a screening source for medicinal chemists for discovering the potential targets. So target, as you have seen in the today's morning session, the target screening is also one of the very important step or basic step in the research stage. So where a lot of companies now they are putting the efforts, because same molecules for the different targets can be the different work for a different indication. So it's a very potential area of research. Efficiently and conveniently obtain the known structural activity relationship of the target and small molecule compounds. So as I told, we have already the journals as well as by assay data and SAR journal data and pharmacological structure data. So this all known molecule structure data is correlated to doing this QSAR studies. To quickly obtain visual clustering analysis of active molecules, skeletons and associated with the target. So this is again, as I told, CyberSAR currently covers the data of more than 10,000 plus targets of 692 species, not only Homo sapiens, but 692 species. In cluster structures, as I told, we have more than 220,000 cluster structures, means the skeletal structures. So which is covers more than like 10 lakh molecules data, and as I told, assay data and assay data, activity data, which also covers the articles and journals for this. Apart from this data, as I told, the pharmacological database structural data as well as patent data. This also has been considered. So in nutshell, targets of small molecules known are clearly visualized, which is greatly improves the efficiency of the medicinal chemist. So medicinal chemist already, if he's just starting the project, or he may be already selected his research area, so it will definitely improves the efficiency of the area of work. So as I told, these are some 4 core functions which I just explained in my previous slide. One is the exploration of potential drug target, and looking for the hit compounds and providing the inspiration for designing first-in-class molecules and facilitating the rapid assay designs and online structural activity relationship. So again, the same. So now this is how the dashboard looks. I will also once end of the session I will show the live website, but this is a screenshot of the website which looks. So this is the target screening prediction. So just if you click on the target, I will show the dashboard in the future slide. Yeah. So these are the three major highlights of our cyber AIDD platform. One is the scaffold cluster and chemical space exploration. So as I told, the core skeletal structure is called scaffold structure, and what is the chemical space for this particular scaffold? You can explore using our database. I think the medicinal chemist is very easy for them to understand. So pharmaceutical target such as tracking index. So if anybody is working for the new targets, and if you are just starting your project, you can input your target here, and based on the our experience, that not the experience means the chemist experience, based on the phase transition success rate, like how many molecules till date have passed from phase 1 to phase 2, phase 2 to phase 3, phase 4. So based on this experience, the AI will analyze and tell you the results, like whether whatever the target you want to work, whether it is of worth or not. So that is one of the good feature which we have, and predict the molecular structure associated with the targets. So this is what I told, the drug repurposing, where most of the companies now are working. So it will predict, already you have a molecule which is you are using like approved for any other indication. So we have the option that predict the new indications for the existing product molecule. Yeah. So this is how the dashboard looks. So if you are like, as I told, the scaffold cluster and chemical space exploration. Once you click any of the target, like example CETP or homocysteine, if I click, there are 75 molecule cluster structures. It has been displayed 75, just the skeletal structures. So inside that cluster, again we have, each cluster have so many real molecules inside. For example, if you choose this cluster, we have 351 real molecules in this single cluster for this target. So similarly, I think 75, and each have so many molecules. So you can just choose one scaffold. I think it will be your PhD thesis. So that is the potential of AI. But just imagine if I want to do it manually, how much time it will take, right? So just for example, if I want to see this structure and search all the journals and all the patents regarding this, and which are the indications it has been approved and which target it covers. So I think this is just 1 minute job in our using the AI tool. So that is the potential of AI tool. And even you can see this cluster, if any molecule is clinical phase, that also you can check. For example, this scaffold, there are 6 molecules which are approved or clinical in the clinical stage, and some molecules are active in this scaffold. Means active means their bio says like more than 10 millimolar, something like that. So I want you to understand how much important in saving the time for your research work. So, for example, this cluster has 70 molecules. The number of original molecules contained in this scaffold. So, like this, 75 scaffolds are there only for one target. So, I think we have more than 10,000 targets, and that number is just like our database is dynamic. We are updating it on daily basis. So, daily we are adding more number of targets, more number of molecules. So, you can just imagine the depth of this tool. So once we deep dive into the real structures, whatever the 70 I told, if you click that, so again all the real structures arranged according to the clinical phase will get it. So whether it is a approved one, or it is a preclinical stage, or the clinical stage, or some are just like bioassay has been published in literature. So that also you will get, like biological assay like 1 millimolar or like that information you will also get. And there is three you can say options here. One is structure information we will get from this, and scaffold information you can get it, and there is an option called bioisosteric replacement. So if you click this bioisosteric replacement, the possible fragment replacement, like if you want to replace any side chain and see what will be the predicted activity, or predicted biological activity, or predicted similarity with the original structure, that also you can check. So later in the demo I will show an example. So this is the assay data. What like it is not that you are just AI is assuming and doing this. So you will come to know the track of the data which has the AI has considered for analyzing your results. So that is what if you click on the CTP and if you click on the chemical space and the molecular structure with the represented value of as I told the 1 micromolar it has shown, and also the respective assay data and respective structure data which are considered to analyze this will also show, which is downloadable. You can take it as a for your reference for your research work. So that is very important. So the molecular cluster associated with the targets and SR analysis, as I told, if you choose, yeah. So once you select that, okay. So as I told, once you choose this real structure, so what are the information we are going to also like trace is. So this is the real structure like which like some molecules are in the phase 3. This data has been collected from data mining. As I told, that AI is not only considering the data from the literature which we are inputting, but there is a team which is doing the data mining from the backend, especially for the molecules which are in the like approved or clinical stage phase, because the data from pharmacopoeia they are doing the data mining, and even for the exploring the molecules, yeah, manual labeling and the data mining. This is as I told, the data mining team will inputting the data. So this I explained already. Based on the phase transition, if you are working on any molecule, any target, if you input here and phase of your project, so it will predict what will be the success for this particular target based on the phase transition success rate. I think if you read any article, if you in the Google, like what is the phase transition success rate, like there is a formula here, simple formula, like phase 1, phase 2, phase 3, phase 4, how many molecules are approved for this target, and what is the success rate from phase 1 to phase 2 with respect to different indications or different diseases. So based on that, it will calculate and give you the what will be the whether your target which you have chosen is a good one or not for your research area. So major highlights, again, the prediction of molecular structure associated with target, as I told, drug repurposing. So here there is an option. So once you go to the real structure, so once you click the predict target, so it will show so many targets, and once you deep dive into your interested target, so it will show like whether that molecule is like how much possibility of percentage which can be like whether that molecule is work for that target or not. So that drug repurposing we call it. So that option. So apart from whatever the few options I told, there are few additional options also, like analysis and exploration of cathode similarity. This is very important for the chemist people. So once you choose any scaffold of your interest, and there is an option called cathode similarity. So it will match with your molecule with its cathode structure and give you the percentage of similarity with whether it is 100% similar or 97% similar. So that time, like whatever the molecule of your design, how much similar to the approved one, that also we can explore. So that is, I think, of very promising. As I told, the commercially viable molecules, if you want to bring to the your research. So another additional function which we can do with this AIDD is like multi-dimensional analysis of targets and indications. So as I told, we have more than 10,000 targets of 692 species and more than 10 million structural data. So we can do the indication associated to the literature and indications associated through the drugs. So AI has the option to analyze, like do the multi-dimensional analysis of the molecule with against the indications as well as the structure. So as I told, there is a linkage between the assay design, target, molecules, and literature. So this you can easily trace with this assay data option. So here is the example, like in one of the target, I have done the assay, when we click the assay data, so we will come to know like where all the our particular molecule of interest is mentioned. As I told, the literature tracking is becomes very important for our research, which has to be published. So definitely the reference is very important. So that is tracked with the this option. So even the proteins, primary amino acid sequence, and three-dimensional structures, because our database is linked to all the databases of public domain like PDB and EDPROT. So there's this all the structures can be traced and it's a downloadable form. So link to the PDB database or for extracting Koch's crystal structure of small molecules and development status can also be traced. So for example, this particular target, what are the molecules till now which are in the clinical, either in the clinical phase or in the approach stage. So that all data we can get it. So again, I'll like in summary, till now what we have seen is, so cybercom has is a molecular generation and evaluation. It is divided into three subsections. That is cyber hit, the discovery of hit compounds, like we have around 10 to the power 64 if you have molecules. So you want to come to the hit compound around 200, then cyber hit is the one. Cyber lead is the generation and evaluation of lead compound, and cyber candidate is the optimization of lead compound and evaluation of druggability. So what are the like advantages? Means the drug molecule design concept is mainly based on the real-time traceable experimental data, not any like only just the journal based data or based on the knowledge. All the tools are based on the real and traceable experimental of marketed drugs. That is very important. So the real-time data is considered for the data analysis. So clinical drug molecules under development. So either approved one or the molecules are in the clinical stage and target active molecular fragments. So this helps to generate the biologically active and patentable chemical space. So this is as the ecosystem in India right now, it is very much favorable for the innovation. So right now this is very good opportunity, because when we were in graduation or in the post graduation, the ecosystem was not so much supported, like funding wise or tools wise, as Sachin was mentioning. In 2008, there was, I think, smartphone use was not that much even, it was not in use, I think, in 2008. Like rarely we used to use. But now the ecosystem is very favorable for the research or innovation. So such tools can be like used for generating the patentable or commercially viable research. So that's what. So using AI algorithms and integration design ideas such as bioisostereochemical substitution, as I had mentioned, so you can replace the site fragments or functional fragments and see the activity. What will be the activity? And now we are also adding one more function, replacing the side chains. Just not just the function group, but also side chains. And next version we are also adding replacing the linker. For example, there are two fragments in the one molecule, and what will be the results if we replace the linker? So there are three more functions which will give the idea for more innovation. So right now the possibilities, it covers 1.4 million fragment replacement relationships forming. So if you replace this 1.4 million fragments, there is a 7 million traceable experimental evidence. So this is a possibility. So advantages, as I told, the database or the tool is based on the real time and data, as well as the one data is of marketed molecules and clinical phase molecules. So when you are considering the successful molecules, there is no chance for failure. Definitely, the molecules which you are getting of, you can say 100% confidence of that there is no These are of commercially viable and patentable ones. So I just read the few important ones that highly human absorb. As I told, we have when we are doing the cyber lead or cyber optimization, there will be some guidelines like druggability, what is the solubility and permeability, toxicity. So the molecule undergoes the screening system so that this sorry so this high synthesizability, because I told the structures of patents also has been considered. So these are of high synthesizability, and safety wise also, like as these are analysis based on the marketed product, so there is no concern of safety. So this is some few examples. So I'll just in 5 minutes I'll just show the some live how it looks. So this is like just I want to end with saying that let the noble thoughts come from all the directions. So we should welcome the new technologies, new ideas, new thoughts, and new updates. Like we should not resist. Try to welcome and learn new things to uh, come up with your new innovation. Yeah, I think it's 5 o'clock. I'll just try to. Yeah, this is how the dashboard looks. So as I told, there are 3 parts. One is CyberSAR. In CyberSAR, again, target overview and cluster structure and real structure tracking. And in CyberX, there are 3 parts. One is hit, lead, and candidate. And there are also some additional functionalities like the virtual library. If some organization want to create in their or database, we can do that. And also there is customized services. For example, if you develop one any hit compounds, around 200 hit compounds you come up with, and you want to evaluate further, we also have some additional functionalities which are inbuilt in this in cooperation with some other organizations. Today morning, I heard from IIT professor that molecular dynamics tool that also is there in this database, but it is like we are working in collaboration with some other organization. So if you come up with your some hit molecules and you want to further evaluate with in incubation with some like universities and all, so we will try to help to do the different analysis. As I told, there is some 40 guidelines, especially in this lead. Yeah. So this is I'm first, once you click on the cyber hit, so here there are 2 options. One is bioisostetic space exploration of your project, and scaffold substituent space exploration. So once you go into the create your project, so there is a option that you can add your own seed molecule or either structure you can draw, or you can write the smells and select the bioisostetic fragment to replace. So you can there is option that you can check the possibility of replacing the isostatic fragment. And once you create this project, so it will give you around uh, out of your this like 10,000 molecules, it will give you 200 molecules. So once you, for example, cyber lead, your target you have to input, and as I was mentioning earlier, so we have around 12,000 structural data in this pharmacological global data set library, and also from the literature SAR data set, it consider evaluate. And this patent SAR data set, it is a like collaborative work, because once you we know that which molecule or which target you are working, the patents particular to that will be analyzed in specific, and that data will be included for this patent SAR analysis. So this is something like what we were discussing with many of the organizations. Like we can have one in silico lab in the particular colleges, and do the collaborative work to bring the commercially viable product. So even if you're having your own data like smiles or it may be that also you can input, and this is very important. So these are some system recommended parameters it will consider to analyze to come up with your lead molecule. This is the one I was mentioning, like complex screening system. So there are 40 guidelines for preclinical development, like it may be ADME, it may be toxicity or solubility, like this. 40 screening systems are there, so which will be analyzed and give your molecule. And even today morning you have seen that professor from IIT. So this is multi-scale molecular dynamics. So this also to be considered. Once he was telling, right, once we get have around 1,000 molecules or 500 molecules, it will be easy to do the molecular dynamics. But if you have 10,000, maybe it is very difficult. Even so, that parameter can also be considered, but that is it's in the collaboration or consultation mode. So once you click that, like create a library and run the module, so you will get 200 lead molecules. So once, I think out of 10 to the power of 64 molecules, once you get 200 molecules, that will be very easy for evaluation. So once you go to the cyber candidate, out of this 200, you can come up with the 4 or 5 lead molecules which can go to the animal studies or preclinical studies or bioassay. So this is a simple dashboard. There is not much complex one. So this is the which I was mentioning about cluster structures. So yeah, so you can click any target of your interest. So this is the one which I showed in the slide, right? So when I click the target, you get 150 results. So out of that, like so many 150 clusters you got, and in each cluster you have so many molecules. So again, you deep dive into each molecules and do the bioisostereochemical replacement and compare the activities. So there is lot to play with the tool. I think once you start using, you can get to know more and more. So that's all from my side today. So any questions from any students or faculty? Okay, thank you very much.
Providing the information regarding how AI tools can be used for innovative drug targets. That's going to introduce the cyber rate. Now I request Nadis sir to illustrate the speaker.
From tomorrow, all the sessions will be held in our college only. And from tomorrow, we start the session at sharp 9:30, and bus will pick you by 8:15, and breakfast will start by 8:30.